Today, I'm going to talk about functional connectivity of the PSTS in the action observation network that is modulated by attention to action features during action and goal inference. First of all, brain regions related to action observation process mentioned usually involves what's called action observation network, which includes PSTS, IFG, and IPL. During action observation, PSTS role is usually linked to perceptual representations of action kinematics, and its response has been shown to be modulated by the viewer's cognitive states. Past studies support the notion that representations in the IFG modulate and restructure perceptual representations in the PSTS via a feedback route through the IPL. However, although IFG has been shown to represent action goals and outcomes during action observations, its function, on the other hand, is heterogeneous and has complex organization. Also, there are multiple pathways proposed for how IFG interact with sensory cortical systems during action recognition. While it is widely agreed that PSTS, IFG, and IPL together support the cognitive process during action observation, it remains unclear how they function as a network during action observation and how they uniquely contribute to this process. Therefore, I will investigate how these regions interact within the AON during action observation and how these connections change depending on the viewer's cognitive states. In the current study, I will characterize the fine-grained structure of functionally connected IFG and other AON regions when attending to action or goal. First, I will subdivide the prefrontal cortex into Brodmann's area. Second, I will identify parcels within IFG and other AON regions that are modulated by cognitive context. Lastly, I will determine which functional networks these modulated areas belong to. In the current study, we have subjects look at a stimuli where a boy or man walk to a closet and perform either one of these actions, jumping or crouching. We directly change subjects' attentional state by asking them to pay attention to different features in the movie, such as what's the actor's action, goal. In addition to the main experiment, we also collect independent localizers to define PSTS in each subject in which they watch a biomotion stimuli. To define ROI used for functional connectivity analysis, I use both a functional localizers and also a data-driven approach to define PSTS, IFG, and IPL. For PSTS, we used a biological motion localizer in which subjects watch patterns of dots moving. Second, because there are no standardized functional localizer to define IFG and IPL, these regions were defined conservatively based on patterns of functional connectivity to the PSTS on the beta series calculated for the main experiment. The detail of the functional connectivity are skipped here, and you can ask me in detail later. And lastly, I divide the IFG and IPL ROI into smaller parcels using the Glasser atlas. And lastly, I identify the functional networks each parcels belong to using the Yale atlas. So here's the results. First, I compared right PSTS connectivity to the right IFG when subjects attend to action, comparing to when they attend to goal. So on the left here, figure 1, you can see that there are a lot more red bars comparing to blue, especially on the right upper corner. This implies that right PSTS and bilateral IFG increased connectivity when attending to action than when subjects attend to goal. However, the right figures shows the IFG univariate bold response is greater during goal rather than action. This is implying that a different neuromechanism involves during attention to action and attention to goal. Further, by investigating the modulated connectivity between parietal and temporal to PSTS, we didn't find a modulation effect on connectivity. However, on the left figure 1, we found out that the parietal univariate bold response was stronger during attention to goal than action, which had been found in previous action observation literature. While on the right figure 2, we didn't find modulated bold response by attention in the temporal cortex. So to quickly recap, we found out that univariate response are stronger during attention to a goal in IFG and parietal cortex, while connectivity between IFG and PSTS is strengthened during attending to action than goal. We further characterize this strengthened connectivity and found out that it's mostly driven by the frontal parietal control network, which is shown on the right figure that in the red color here. This implicates that attention to action strengthens a selective pathway within the action observation network. Similarly, we want to look at this selective pathway between IFG, parietal, and temporal as well. Here, you can see that most of the modulated connectivity between the IFG are between the IFG and temporal cortex. Further, comparing the left figure to the right, we can see that the functional system involved are dependent on whether subjects attend to action or goal. And lastly, these modulated connectivity are mostly between the frontal control network and default mode network, as shown in the figure here as the colored pathway. So in summary, we found out that the communication between PSTS and IFG is strengthened in the frontal parietal executive system when subjects attend to action than when they attend to goal. And also by comparing both univariate results and connectivity results, we found that the modulated connectivity are distinct from neural populations representing action goals in parietal cortex. And lastly, by further characterizing the modulated connectivity between IFG, temporal, current study provides a possible evidence for a ventral pathway between IFG and temporal cortex for attention modulation during action observation.